Thank you, Professor. Good afternoon, everyone. My name is Emmelyn Hsieh, and this summer I worked in Professor George Brooks's exercise physiology lab on a project that looked into supporting brain glucose metabolism in traumatic brain injury patients. So what exactly is traumatic brain injury? Traumatic brain injury, also known as TBI, is a brain injury caused by a bump, blow, or jolt to the head, or a penetrating head injury that disrupts the normal function of the brain. So there are three classifications of traumatic brain injury, from mild to moderate and to severe, but our particular study that I'll be talking about today focuses on severe TBI patients. As of 2011, there were approximately 1.7 million traumatic brain injury cases recorded annually in the USA, and the Center for Disease and Control has recorded some more specific statistics on those cases. So out of those 1.7 million cases, about 1 million were emergency department visits, 275,000 were hospitalizations, and 52,000 resulted in death. To emphasize the lasting effect of TBI a bit more, about 5.3 million people were reported as patients living with a traumatic brain injury-related disability. So evidently, traumatic brain injury cases represent a large number of the patients that visit the hospital and that need intensive care. So our project today serves to look further into these patients' bodily functions to provide some grounds for future research into better forms of treatment. So what do we know about the brain? The brain is an organ that relies heavily on glucose, and a healthy brain with no damage will use this glucose to generate adenosine triphosphate, also known as ATP, which is responsible for energy transfer. So as seen in this simplified diagram for a healthy patient, the breakdown of glucose and release of ATP is accomplished through these three following metabolic processes, from glycolysis to the Krebs cycle and on to the electron transport chain. On the other hand, for a traumatic brain injury, or TBI patient, you can see that our very first metabolic process, glycolysis, is now being blocked, which prevents any ATP production or breakdown of glucose. So we know from previous studies that there's evidence of less glucose uptake in the brain, and this is actually a classic indication of glycolysis blockage. So although we don't know exactly what step of glycolysis is being blocked, we do know that it is this exact first metabolic process that is causing problems within our traumatic brain injury patient. So this is a very new field of research, and there currently have been no studies done that have looked into the whole body glucose metabolism in traumatic brain injury patients. And knowing all of this led us to develop our hypothesis for this project. So we postulated that traumatic brain injury patients at rest will demonstrate impaired glucose metabolism compared to our healthy patients at rest. Seeing any differences when comparing our healthy patients to our TBI patients could demonstrate any number of problems in the TBI patients, from glucose uptake to glucose clearance and even glucose production. So a brief overview of the setup. We had 20 total traumatic brain injury patients evaluated in this project, and the researchers at UCLA Brain Injury Research Center gave each patient a continuous intravenous infusion of deuterium and 13C labeled glucose, as pictured here. As you can see on the left-hand side molecule, the red circle indicates where a 13C has replaced, normally there would be a 12 carbon there, and on the right-hand side molecule, the red circles indicate where deuterium has now replaced the hydrogens. The amount infused, however, was a very small percentage, and not so much that it changed the body's natural glucose concentration. So why do we use isotopes in this experiment? The reason we use isotopes in the body is that the difference in weight of these labeled molecules from the natural molecules found in the body will allow us to track how they are being used by the traumatic brain injury patient. So this is a simplified diagram showing what happens to our two isotopes. Both of our labeled glucose molecules will undergo the same metabolic processes that I mentioned before. However, all of our deuterium molecules are eventually lost and released in the form of body water. The red circle here highlights the movement of the deuterium from our isotope into the into the water molecule. On the other hand, some of our 13C molecules can be recycled by the liver and will then re-enter the blood as newly formed glucose through a process called gluconeogenesis. The researchers at UCLA then took blood samples at 4 time points with an hour in between each sampling. So for example, T0 would be before we started our infusions, T1 would be 60 minutes into our infusions, T2 120, and T3 at 180 minutes for a total of 3 hours of infusions. They then added chlorhydric acid to stop any further glycolysis and then centrifuged the blood samples to isolate the plasma, which then the plasma was frozen into tubes to be sent to our lab for analysis. So this is just a simple overview of the processes we use to isolate the glucose and run it through our equipment. We started off with our first step, ion exchange, which separates the glucose from everything else in the plasma based on its charge. In the second step, lyophilization, that was what we used to dry the samples. And in the third step, glucose derivatization, what it does is it stabilizes the glucose molecule so that our equipment can read it. Our equipment, called gas chromatography mass spectrometry, or GCMS, is pictured here in the photo right next to me. And what the equipment is able to do is it separates the molecules by weight, and then we're able to specify ion fragments that the GCMS can count according to their mass to charge ratio. Eventually, the GCMS would then give us some data that we could evaluate as a spectrum, as shown here. As you can see on the label in the left-hand corner, the x-axis represents the time that the fragment was eluted, and the y-axis represents the abundance, or basically how much of that specific ion fragment the GCMS was able to count. So from seeing these abundance values, we were then able to determine the isotopic enrichment of our two tracers, and therefore calculate our glucose turnover rates and get an idea on the whole body glucose metabolism of our TBI patient. So now let's look at some of the results that we got. When we compared the average glucose concentrations of our traumatic brain injury patients to the historical data of our healthy patients, we did not see a significant difference, as you can tell from how close the bars and values here are on the graph. However, as you can see from the red circle I put on the right-hand bar, the variability of the data for our traumatic brain injury population was much greater than for the healthy population. So in other words, in the healthy patient population, they could have seen a pretty consistent average of about 5 millimolars for glucose concentration, but when we looked at our TBI patients, we could have seen glucose concentrations from as low as 2 millimolars to as high as 10 millimolars. In the body, isotopes are naturally occurring, and deuterium has a natural occurrence of 0.015% of all hydrogen atoms, while 13C has a natural occurrence of 1.11%. So as you can see from the graph on the left-hand side, the 13C glucose line is continuously greater than the deuterium glucose line across all of our time points. Again, I'd like to mention that the two tracers we used are both broken down the same way. However, the deuterium glucose line, the deuterium is lost as body water, while the 13C can be recycled by the liver. So the key to this graph we have here is that the differences between our two tracers and the rates at which they are being used actually represent our glucose recycling rate and can give us an estimate of the body's gluconeogenesis or glucose production. For our rate of disappearance, we ran a t-test on our data and found that when using the 13C labeled glucose to calculate rate of disappearance, it appears to be significantly lower than when we used D2 labeled glucose to calculate the same rate. The disappearance of deuterium-labeled glucose actually represents the body's true utilization of glucose. So essentially, you can think of it in the sense that the 13C label reappears in the newly formed glucose to replace whatever was used up due to glucose recycling. So it looks like we actually lost a lot less 13C glucose. Therefore, again, the difference between these two bars is also because of glucose recycling. For our glucose recycling rate, we were able to use the tracer isotopic enrichment that I mentioned from before to estimate gluconeogenesis. Gluconeogenesis is just one of the ways the body produces glucose. And as fasting continues, gluconeogenesis plays an even greater role. So this is evident from the differences in the 3 bars of the graph shown here. On the very left-hand side bar, the historical data of healthy patients shows that after 2 hours of fasting, gluconeogenesis provides 20% of total glucose production. In the middle bar, you can see that after 12 hours of fasting, gluconeogenesis increases to 40% of total glucose production. And on the very right-hand side, for our TBI patients, who were fed intravenously and were actually not starving, we found gluconeogenesis to be greater than 50% of total glucose production. So there's a clear trend here for the TBI patients to have about a 30% greater gluconeogenesis than the healthy patients fasted for 12 hours, and 170% greater gluconeogenesis than our healthy patients fasted for 2 hours. These observations help indicate that the TBI patient's brain thinks the body is starving and therefore responds accordingly by ramping up the gluconeogenesis in the body. To summarize our findings, we saw that the glucose concentrations in the two different populations were not very different, although the traumatic brain injury population had much more variability in its glucose concentrations. We also observed a greater isotopic enrichment in 13C glucose when compared to D2 glucose, and used these to estimate our whole body glucose metabolism. In addition, we noted that the rate of disappearance when using 13C labeled glucose was calculated to be much lower than when we used D2 labeled glucose. And last but not least, our glucose recycling rate averages were seen to be different, and we saw a trend for our traumatic brain injury patients to have a 30% and even 170% greater gluconeogenesis when compared to two different sets of historical healthy patient data. So seeing the TBI patients show that gluconeogenesis provides such a high percentage of glucose production is actually an incredibly important finding from our study. Our TBI patients are being fed, unlike the fasted healthy patients that I mentioned before. So they actually do have glucose circulating in their body, and they are not starving. However, the brain seems to perceive that the body is starving because of its glycolysis blockage, and so responds by increasing gluconeogenesis. And this leads into a hypothesis for some possible future studies. It's important to note that in the process of glycolysis, the glucose molecule is broken down into pyruvate, and then can either go into the Krebs cycle or can be converted into lactate by an enzyme called lactate dehydrogenase, also known as LDH. This process of converting pyruvate into lactate is actually a reversible reaction. So essentially giving a patient some lactate could give that patient the chance to convert the lactate into pyruvate and bypass that first blocked glycolysis step to continue the metabolic processes and create energy. So we have sufficient evidence from previous research to know that lactate in the brain can be efficiently and effectively transported into the brain's mitochondria. So these facts lead us to wonder, can the human brain utilize lactate just as effectively as it does glucose? And if so, can the lactate be used as a treatment for future traumatic brain injury patients? If this is indeed possible, then patients could potentially recover much faster and reduce the amount of brain and bodily damage that comes from traumatic brain injury, since they would now have an alternative source of energy for the brain. This concludes my presentation. I would like to thank Dr. George Brooks, Dr. Hsieh and Mike Horning for being such great mentors. I would like to thank the UCLA Brain Injury Research Center for working with us, and I would especially like to thank Nathan Hsieh, the SURF program, and the Rose Hills Foundation for giving me this opportunity this summer. Thank you all for listening, and does anyone have any questions for me to answer at this time?